So Andrea did some homework during the break, and so she's going to tell us about centromeres.
Okay, so we talked about those gaps, those funny gaps on chromosome 1 and 9 in the Manhattan plot, and Patrick was right in his identification that they are centromeres, which is the middle bit of the chromosome when the cell divides. That's where all of the scaffolding attaches. And the centromeres tend to contain a lot of repetitive elements. They tend to have the amount of repetitiveness varies from chromosome to chromosome, and there are 3 big chunks of the human genome that aren't actually fully mapped yet. And the big chunks are the centromeres of chromosome 1, the centromeres of chromosome 9, and a big chunk on the Y. But because we don't usually put the Y on our GWAS plots, it doesn't show up as a gap. There will be gaps, though there are smaller gaps on all of the chromosomes. So if we zoomed in further, you would see a little gap like that on every single one of the chromosomes because they all have the same. That's me.
Cool. Great. So I'm going to talk to you guys before lunch about winner's curse and population stratification. And so first we're going to start, so just to, we're going to talk first about winner's curse. We're going to talk about how to deal with it, how to correct for winner's curse. Then we'll have a discussion of population stratification, and then measuring it if it's there, and how to account for it if it's in your data. I have some supplementary slides at the end. We probably won't look at them, but you'll have them available to you. I'm gonna rush through this just a little bit in the interest of time, and so well, you'll see. So I'm going to try to rush through this so we can spend a bit more time on population stratification, which is, I think, a bit more central in the sorts of things that we run into. And so I want to first talk to you about the man flu. So I eat way too much junk food, and so I'm ill more often than I should be. And so I'll feel sick in the morning, and I'll tell my husband, oh, I think I have the flu. I'm doing really poorly. And so he takes my temperature. And if my temperature is like 99, he's like, you don't have the flu. You're fine. But then sometimes I do. Like sometimes it is like 102.5, something like that. These are Fahrenheit temperatures, but that's high, right? And so then it's like, okay, if my temperature is high enough, he's like, okay, maybe you do have the flu because you have this really high temperature. So let's say I started recording every time. It's like, okay, John says I have the flu, it's okay, and I write down the temperature that day, and I do it every time he gives me the thumbs up. And then I take the average of those because I want to know, well, what is the average temperature of me when I have the flu? What's going to be the problem there? There's going to be some bias that gets introduced based on the sampling mechanism. What's the problem? Well, when you have the flu, there is some variance in your temperature, right? And so some of the time, when I have a temperature 98, maybe I actually do have the flu, right? And so what is that going to, how will that affect our estimates of the average temperature of a person with the flu? It'll be biased upwards because we're throwing out all of the data when it's too small. And so this is related to just general work that we do. So when we take an OLS estimate, like Dan was showing in our GWAS examples, we're using OLS or something kind of like OLS. And OLS, one of the nice things about it is that it produces unbiased estimates. And so we should feel really lucky about that. A problem that we generally see in research, though, that use these data is this is just an empirical point. There were a lot of these studies done, and they find these big effect sizes in their discovery sample, and they take it into their replication sample, and all of a sudden, these big effects that they saw in discovery were much smaller in the replication. And so this phenomenon that's just seen empirically is called the winner's curse. The issue, I'm going to give you the underlying issue of why this happens is because you shouldn't be using the same data set for selection of your SNPs or of whatever factors you're interested in, and for estimating effect sizes. And I'm going to show you why, but that's kind of the overarching principle. It's also related to regression to the mean. On average, we think things are small, but we sometimes just see big things by chance. This is relevant for all the research that you do. So you should keep in mind that if you're using the same data to make your big discoveries that will make you famous, as you do to estimate how big those effects are, well, given that we kind of discount things that are insignificant, which means things that have small effect size, that we estimate with small effect sizes, we're going to have this winner's curse problem generally. So, we're focused on GWAS and genetic stuff here, but we should think about this in everything that we do. So there's kind of two ways in GWAS that we might run into winner's curse problems. There's kind of two selection mechanisms that might cause us problems. These aren't like technical terms, but this is how I think about them. We have selection by threshold and by rank. And so for threshold, Dan already told you, we're doing a GWAS. We only want to have SNPs that are significant. And so we say, I'm only going to look at SNPs that have a p-value of less than 5 times 10 to the minus 8. Well, for a given sample size and for a given standard errors, that means I'm actually going to only take SNPs that have an effect size, a beta hat, greater than some value. And we're back in this flu world that I told you about. And so there's some threshold that we throw out any information or any SNPs, or we ignore. We ignore any SNPs that don't have large effect size, but we just look at the big ones. So the threshold is great for multiple testing. We talked about how we want to have this really tight threshold for multiple testing, but it's going to create problems for the effect size estimation. And so I have this picture here to illustrate why this bias occurs. And so on the x-axis, we have the beta hat estimate. And so these estimates correspond, let's say that we had a true effect size of exactly 0.1. So that's the ground truth. And we have a sample of about 100 people, right? So if we just did a GWAS and if we just did an OLS estimate and we want to say what's the distribution of the beta hats that we'd get if the true beta is 0.1, well, it's going to be a normal distribution centered at the truth, right? But if we're conditioning on it being significant, then that means I'm going to just throw out anything that is less than negative 0.2 or greater than negative 0.2 and less than positive 0.2. And so I'm deleting all of that. And so the new distribution has this big chunk out in the middle. These get inflated just so that it integrates to 1. But you can kind of see what's going to be the mean of a random variable that has a distribution like this. Like, who thinks the mean is going to go this way? Who thinks it's going to go that way? Yeah, right. Yeah, well, most of you. Who thinks this? You didn't vote. Who thinks it's going up? Yeah. Okay, yeah, but you're right. You can kind of see there's just so much mass over here that this is going to drive our mean to the left. And that's the source of the bias from winner's curse. And if you imagine that the true effect were way out here, right? Like maybe it was out here, and so this normal distribution was living on this side. The chunk that's getting cut out would be quite small, and so it's not going to drive the bias a lot. And so in general, we think that the winner's curse is going to cause a bigger problem when the true effect lives near the threshold that we're going to use to identify significance, and it's not quite as big a deal when we're very distant from the threshold. So that's just some intuition. Yes?
Is that not very similar to the idea of training validation testing in machine learning, for example? Which is like using training to get your estimates done.
Yeah, it's it feels related. I don't know exactly what but yeah, it is true. You want to use different data for some of these different kind of tasks, like for identifying the instruments that are going to go into your machine learning something, and then testing train. Yeah.
Yep.
So yeah, it's I think it's exactly related. Mm-hmm.
To focus this a little bit, the reason why we care about it maybe a little bit more in the context of genetic data is that because the effect sizes are really small, and then we are actually in the domain where we're not really removed from the threshold. We're going to have more winners first when the effects are so small, and that's why maybe we should think about it in this context.
Yeah, I mean, it tends to have more to do with how close we are to the threshold. Like, I mean, the effects are small, but if our sample sizes are like 80 bazillion,
yeah, that was my next question.
Yeah, then winner's curse won't be as big a deal as sample sizes get really large.
So, so you don't think of it as a special problem in the context of genetic data, but just in general something we need to think about?
I'd say, yeah, it's generally any time that we think that we're just barely well paired, and this goes back to the stuff that Dan was showing with the type M errors. Like how much inflation do we expect to see in our estimates? Well, we saw when we were low powered, meaning that the truth is probably close to this threshold. So like if the truth were right like exactly at the threshold, that corresponds to roughly 50% power, right? And so and the further we get out from the greater the power, it means that the true effect is further and further from our critical value, and so our type M error is gonna go down. So it's the same phenomenon that Dan showed you yesterday. You happy? Great. And so here's another type of selection by rank. And so this goes back, Dan said he was going to punt, but I'm going to give you the super brief version. When we're picking the lead SNPs, so when we say like, what's the SNP in the locus that we're going to call like the important one, what we essentially do is we just take the SNP in a region with the lowest p-value. And we say that's the lead SNP. And so by that, we not only have a selection by thresholding, we're only choosing things that are genome-wide significant, but of the SNPs that are in a certain region, we're taking the most significant one, which will roughly correspond to the one with the biggest effect size. And so we have this kind of rank selection as well. I don't know of anyone who actually addresses these rank selection issues. It's a bit more complicated because order statistics and stuff are a bit more of a pain to deal with. But this is something you should be aware is there. Yeah, I can't think of any paper I've ever seen that address, any kind of GWAS type paper that addresses this issue other than saying, oh yeah, there's winner's curse because of something like this. And so how do we deal with the winner's curse? Well, we could replicate, right? So if we have our one data set, so going back to the machine learning, we have our training data set, and then we can take it into to the testing data set, and we can replicate it. And because OLS is unbiased, our replication data should and we're not gonna do any thresholding in the replication stage. We're just gonna look at what the betas are. Replication's a way that we could maybe solve winner's curse problems. It's a bit expensive because you have to set aside replication data. And so, we may or may not want to do that. We may or may not be able to do that if we don't have access to the individual level data. But this is a pretty clean way to deal with winner's curse problems. There's another ways that you could just try to correct for the winner's curse. I'm not going to really talk about these much in the interest of time. There's readings that you could look at if you want to. I'm going to talk super briefly about, you could do a Bayesian correction, which is what we end up doing in a lot of our GWAS studies in the SSGAC. And the idea is pretty simple, right? We want to know what the distribution is of the true effect given the observed effect and that it's significant. Right? And so this is the thing that we want to know. We could take the expected value of that thing if we knew this distribution. Well, this is a conditional thing, and so we could use Bayes' rule. We'd end up over here. I can remove significant because conditional on knowing the beta hat, we know already whether or not it's significant. And so I just remove it there and then replace it with this. Well, this term here's the likelihood. It's the distribution of the beta hat. Like if we knew what the true beta was, how would the beta hats be distributed? Well, that's easy. It's just going to be a normally distributed thing with mean whatever the true beta is, and variance equal to the our standard error squared, right? So that's not so hard. But we also have this prior. So what's the distribution of the betas, genome wide that we have in the data? And so that's a choice we end up needing to make. And so if you wanted, you could assume it's just Gaussian. That's really easy and clean, because this is normal and this is normal, and so they're conjugate and everything's pretty. But not necessarily realistic, right? We don't, if the betas are actually Gaussian, that's implicitly assuming that every single SNP affects or is associated with the phenotype we're looking at. And we're talking a little bit about that. That's not quite right. So what we might want to do is say, okay, well, let's say it's normal, except that we'll do a mixture distribution where 70% of SNPs are null. They have exactly zero effect, but conditional on not being null. Maybe they're normally distributed. So that feels a bit more realistic, perhaps. And so we could use a distribution like that if we wanted to try to plug it into this equation to do our correction. And so this is not crazy. It seems to work pretty well in practice. I don't want to talk a ton about this other than to point out that no matter what correct, oh, sorry.
Yeah, just a quick question. That's not equivalent to some sort of shrinking?
It's going to ultimately be shrinkage, right? Like it's gonna look just like any of these shrinkage type estimators.
Can we just do that in the first step? Like instead of just running the OLS, putting in some random effects for chromosomes or something like that? Because you are just choosing the leading SNP within a chromosome that is combining all the effects. So if you shrink the first stage, want to have that same.
Sure. I mean, like, yeah, you could in theory. I mean, the reason that we don't is a bit just based on history. Like, what's traditionally done. Okay, well, there's a couple of reasons why it's tricky. So traditionally, GWAS research was done by asking a bunch of cohorts to each run their analysis and then meta-analyzing together, right? Whereas these shrinkage type estimators usually require the individual level data. And so I guess you could have each of them like run a shrinkage estimator and then meta-analyze those. But the problem there is that the amount of shrinkage that you do is usually a function of the sample size that you have. So if you have a very small data set, then you shrink a lot because there's a lot of noise. And so then you end up meta-analyzing a bunch of things that have been over-shrunk. So I mean, it's tricky. Like you
analysis is itself shrinking, so it's like.
It's shrinking. If the means is the same across the cohorts, it's not shrinking, right? They're all mean beta, so that it's making things more precise, but not necessarily shrinking towards zero like a shrinkage estimator would. So like, yeah, I mean, to answer your question generally, like, yes, you could, but no, we don't, based on data limitations and historical sorts of things. Yeah. So I just want to make a little point here. So I'm saying we're doing a correction. There's this result from this paper here saying that no matter what correction function we use, there is no correction function that's going to produce unbiased beta estimates for all beta, which just means that there's no ideal function that will fix all of the bias in our estimates, no matter what we do. And so you should be skeptical of any kind of correction for winner's curse that people do for individual SNPs. We can hope that maybe there's less bias, and we can also hope that maybe on average the amount of bias is fine. Some of the shrinkage will shrink it too much, and some it won't shrink it enough. But maybe on average across the whole distribution of the beta hats, things are okay. But yeah, just a small point. So here's.
And that should be true if you did a Bayesian approach with the correct prior, right?
Yeah. So you'd hope. Although I used to have a line in there about that, but then I realized it's not quite precise. Yeah, we can talk about it maybe after. I had the sentiment, the sense that if you're doing this Bayesian approach, then you will on average be right, meaning that the bias will be mean zero genome wide, and that's true. But one of the reasons that it's true is because we have a symmetric distribution. And so, if the betas that are like +1 are offset by the betas that are -1. So it's also true that if we shrunk everything to exactly zero, it would be on average right. And that's not good. Any case, that's a side note.
So one of the things that you're trying to do, obviously, is you're trying to sort of fill out that kind of, the slab that was taken out because of the cutoff. If you could, is there any advantage to doing like subsampling of the population and sort of getting a distribution of the beta values by doing subsets of the population and running regression on a SNP by SNP basis?
So, I mean, it sounds a little bit like you're describing something that's a complicated version of replication. Is that like?
Essentially, but it's using your sample. Just say you're. I mean, I understand that it cuts into power a little bit, but if you could sort of redo your regression on a SNP-by-SNP basis and get sort of distribution of betas based on what kind of on a smaller subset, like a random sampling of all.
Yeah. I mean, there are probably things that you could do that, that are replication-like, that are higher powered than actually just holding something out. Okay. But I don't know that I know of people doing that. I'm not exactly sure how you do it, but I believe that something like that could be done. So here's a quick example of a replication and correction that we have from the EA. I guess this is EA1. So EA1 we had three hits, and so these were the naive, like the uncorrected estimates that we had for those hits. So these are the actual estimated effect sizes, right? We pass it through this Bayesian correction machinery that I told you about, and you can see that these always get a bit smaller, about 40% smaller. So we go from 0.1 to 0.06. Here we go from 014 to 009. So all of these are shrinking to account for the winner's curse. And how well does that do? Well, we had a replication sample, and we can see how close our replication is to our corrected estimates. And it looks like for this SNP, it maybe helped. We dropped from 0.1 to 0.07, and we guessed it was going to be 0.06. These other two don't look as they're good. Although, looking at the standard errors here, these are all quite noisy. So I don't know how much we can learn from this, but we do generally see that the replication, in 2 of the 3 cases, the replication estimate is smaller than the discovery estimate, and maybe we're pretty close here. I don't know. But we don't know that these individual SNP estimates are incredibly reliable, and so maybe if we do something that's a bit more genome-wide, we look at a broader range of effect size estimates, that would be more interesting. And so one place where this can become important is when we're doing power calculations. So let's say that we do our GWAS, we get 100 genome-wide significant loci in our GWAS, and we want to do a power calculation to figure out how big of a replication sample we'd need to be able to detect these SNPs in replication. Or alternatively, we have a replication sample of a fixed size, and we want to know how many SNPs do we expect to replicate given the effect sizes that we've observed and the effect sizes that we've observed and the sample size we have available in replication. So if we didn't correct for winner's curse and we did a power calculation, how is that going to affect our power calculation? How is that going to get us in trouble? Yeah. And so then what would that mean then if we think our power is too big? Exactly. So our betas are too big, so we're going to say, oh, we have lots of power, we should expect all of these to replicate. Or in the other situation where we're trying to choose the replication sample size, we would say, oh, I only need 5,000 people to be well powered in my replication, but that's based on estimates that are much too big, and you actually maybe need a sample size twice as large. And so if we don't do any kind of correction like this, or we don't take the winner's curse into account, we can make big mistakes in our power calculations that will cause us to think we're well powered when we aren't well powered. And so how am I doing on time? I think I'm not going to go into close detail on exactly how we do these power calculations, but I'll give you kind of the overarching idea. So what we did is so for Akbe et al., we had 74 genome-wide significant hits. We did this Bayesian correction, and so then for each SNP, we knew what the distribution of what our posterior distribution was of the effect sizes. And so what we did is we drew random draws from the posterior distribution for each SNP. We added noise corresponding to how much noise we'd expect to be for a replication sample of a certain size, and then we looked to see how many SNPs were significant in this simulation. We did this over and over and over again. So that was the idea behind how we calculated power. And so when we did this simulation to look at how many SNPs we expected to replicate at 0.05, we guessed that there would be 40.3 out of the 74, and in the end we saw 52. So this was based on a Gaussian. I told you we have to make a choice about a prior. So this was based on a Gaussian prior, and we did even better than expected in this case. So that was nice. For the Akbe et al. Subjective well-being, the GWAS of subjective well-being, we had 19 genome-wide significant hits. We passed it through our machinery to calculate power, and we expected about 16.7 of them to be replicated at a p-value of 0.05. And then when we did our replication, we got 16 out of the 19. And so it seemed to work pretty well here. In this case, we used a spike and slab prior. I don't know the degree to which we're a bit closer here than we were here, and I don't know how much of that has to do with having the better prior versus just being lucky. But this seems to work pretty well, is what I'm saying, at least in these cases. If you want to have read into more detail of exactly how this is, there's pretty careful write-ups in the supplement of this analysis. So if you wanted to use something like this in your work. Happy? Okay, great. So for winner's curse, so the big point is that we shouldn't use the same data set for discovery as we do for sample size estimation, or if we use the same samples for that, we should understand that our estimates may be biased upwards or away from zero due to Winner's curse. There are approaches that allow us to correct for this bias with limitations, though. Okay, so that's all I want to say about, well, that's all I plan to say about Winner's curse. Do other people have questions? Yes?
Can you explain a little bit? So you're correcting betas, but how do standard errors enter into that, if at all? Like you can have a high beta and low standard error, and does it help in any way? Or you can have really high beta and really high standard error.
Yeah, that's a great question. So if you have a large beta and a small standard error, so a small standard error means that the critical value, like the threshold, is going to be quite small. And so if the true beta is very large relative to what the critical value is going to be, then we're going to be very distant from the threshold, and so the Winner's curse probably isn't going to be that big a deal. On the other hand, if you have a very big standard error and so that our true effect size is kind of near the threshold, we're going to see much more inflation relative to the estimated effect size. Does that?
So those betas just not going to get corrected much?
Yeah. So if we were to draw, so we could draw like a correction function, so like this is like beta corrected, and this is our beta hat. So if we didn't do any correction at all, we'd be on the 45-degree line, right? And what the Bayesian function, if we were to map this out, it tends to look something like this. And so you can sort of see up here, we really don't correct the big things, but when we get down here, we shrink them a lot. Great. Yep.
A related point, though, I think that in addition to the betas being inflated when you find something significant, the standard errors are underestimated on average. Isn't that right?
Why?
Because these are things that happen to be significant, and that could be a draw of a low standard error estimate relative to the truth.
That's true. So I guess we don't often think about standard errors as random variables, but they are. And so so it could just be that we have a low, but that would be then taken into account by, using a t instead of like a t-score rather than a z-score, right? Because that accounts for the randomness of the standard error. So as long as the critical value, I guess I have to think through. All of the math that we've used to do these assumes that the standard errors are, well, no, because we use it as a function of like n and stuff. I'd have to think about it. That's a hard question. I think it's going to be somewhat answered by using t statistics instead of z statistics, but maybe not entirely. Okay, great. So let's talk about stratification then. So we've talked about, we've mentioned it a bunch of times since we've gotten started, and I think we're all aware that it's a major source of bias in the problems we have. So just to give some intuition of what stratification is. So the classic example of stratification, there's a 1994 paper called the chopstick gene. I'm going to change it a little bit. So I'm from Utah. And Utah has the highest per capita Jell-O consumption in the country. And I fit right in. I love Jell-O. And I love it so much that, let's say I decide that, this is more than just environmental. There must be some biological underpinnings to how much I love Jell-O. And so I'm going to do a GWAS, okay? So let's say that I hired one of you as my GWAS consultant, and I approach you and I say, hey, I've gathered some data on Jell-O consumption. I've asked a bunch of people how much they consume Jell-O. I've genotyped them all. What's the first thing you're going to ask me when I tell you, okay, help me out. I don't know anything about GWAS, but I have some data. How much do you have? Yes. so much, because you're worth it. What's the next question? So what are the important things that we talked about with Dan? Sample size. Yeah. Okay. So I say I've gathered data from 50 people and I want to do my GWAS, and you say no. I talked to Dan, and Dan said that's a bad idea. Okay. So then I say okay, great. So I listen to my consultant. I go out and I gather data on 80 million people spread out all over the United States, and I do our power calculations, and I find out that, I have 100% power to detect a bunch of SNPs at a p-value of 0.05. Okay? Then what do you tell me? Am I good? No, why not?
Your p-value's off.
Yeah, multiple testing, right? So those are the first two big, you were listening to Dan. He likes this. So you need to worry about power, we need to worry about multiple testing correction, okay? So the next thing I tell you is, okay, I use 10 to the minus 8. I create a Manhattan plot, and I still find a bunch of really strong hits, Now, do we think these hits have biological significance, given what I told you at the beginning about Jell-O consumption in the United States? Probably not, right? And so it might be that there are certain SNPs that are just more common in Utah for some reason, just by chance. And there's environmental things, I don't know what those are exactly, but that just cause us to eat more Jell-O there. And so we find all these hits just related to the fact that this is where you're from geographically, and there's slight differences in allele frequencies across the country. And so that's the general idea of what stratification is and why it's a problem. So let's put some math behind it. So remember, this is the model that Dan proposed. It's we have some phenotype Y, so it could be education or whatever Jell-O, and then we have a bunch of SNPs, right? So this is the additive factor from the model that Dan showed, and we have some residual here. But I've added this G term. I guess I should have maybe made this the Z gamma. It's kind of standing in as that if we wanted to. Dan told me I should harmonize notation. I guess I missed that one. But this is supposed to be some sort of fixed effect factor that correlates with the person's ancestry that affects Y. And so this could be kind of where you're from, what your ancestry is, anything that may be correlated with your genotype. And so the assumptions that I'm going to make here, first I'm going to start and say let's standardize Y and X, so they're mean 0 and variance 1. And we're going to do that just to make the algebra a bit easier. So that's not super important. We're going to allow for the X's to be correlated. So people's genes are correlated. We've talked about LD a bunch already. And so I'm just going to allow there to be LD. We're good. But I'm also going to allow the X's to be correlated with the G's. And so, in the chopstick example, that might arise because, there are SNPs that are more common in Asia. And people in Asia tend to more often use chopsticks, and so that'll induce some correlation. So those are allowed to be correlated, but this residual piece here is going to be uncorrelated with everything else. So here's the framework that we're going to use to talk about stratification. Yeah.
But given the idea of there's maybe more SNPs found in Asia and people use chopsticks more often in Asia, it doesn't seem there's a connection between distance and chopsticks. Is that what we're trying to?
That's the whole point.
Exactly.
Yeah. Yeah, and that's what we want to avoid, right? Because we're hoping that these SNPs have to do with something that has its roots in the SNP itself, right? And if it's just because the SNP happens to be more common someplace, then then that's not really the start of the not the start of the mechanism. Mm-hmm. Okay, great. So I told you that SNPs can be correlated, but SNPs can be correlated for a bunch of different reasons. I'm going to talk about two primary reasons. And so one is linkage, and the other one is population structure, right? So linkage we've talked about a bunch. We have SNPs that are inherited together when they're on the same chromosome. And because they're inherited together, they tend to be correlated with each other, because if you get one, it comes with the other, unless there's like a recombination event between them. And so the correlation of a pair of SNPs due to linkage tends to decay with distance So if you're on the same chromosome but you're very far apart, then you're less likely to be linked. And if you're on different chromosomes, then you're definitely not linked because chromosomes are inherited separately, right? So the amount of correlation between SNPs that's due to linkage is pretty predictable and geographic, right? But there also can be correlation due to population structure for the reasons that we talked about. So let's say that we have a pair of SNPs that are much more common in our European sample and less common in our Asian sample. Well, if I told you that, that I had a certain version of SNP1, then not only does that tell you about just what my genotype is, but it means that I'm a lot more likely to be in this group because the SNP is more common in this group. And because it's much more common in this group, it means that I'm more likely also to have SNP2. Right? And so that's going to induce correlation due to the population structure. And so generally correlation between pairs of SNPs, the language is a bit fuzzy, like even among, when I'm talking to my colleagues, like geneticist colleagues and stuff, they tend to be a little loose in the language. Linkage disequilibrium is technically just any correlation between pairs of SNPs. Sometimes you'll hear people use it to just correspond to correlation due to linkage. So, it can get a little confusing, but that's just to be a bit precise in the language. But both of these are sources of correlation in our data.
So what degree do LD and population structure overlap? Is it the case that if we sort of look closely enough that there might be different LD patterns in people from Utah compared to people from other states?
I will talk about that on Monday.
Great. Yeah. I'm going to assume generally, though, that the LD, or the correlation due to these two factors, are separable. And under pretty reasonable assumptions, I think this is fair. If
there's inbreeding in the population.
Yeah. So this is why I used to just say there are two reasons, and I've changed it to two primary reasons. So like things like assortative mating, or even just like technical errors, like what chip you're, like batch effects and things. There's lots of things that can induce correlation that are also problematic. I'm not going to talk about those right now. We are going to talk about a lot of these later, but just for the sake of this lecture, I'm just going to say these are the two main ones. Great. And so if we take the covariance between two SNPs, we can say, well, there's some of the covariance due to linkage and some of the covariance due to population structure. Okay? So going back, so when we do a GWAS, as Dan told you, what we're doing is we're regressing the phenotype on a single SNP, just one at a time throughout the genome. And so for a simple regression, if you remember your basic stats, a simple regression, the actual coefficient is equal to the covariance of our two, of the right and left-hand side variable divided by the variance of the right-hand side. We've assumed that this is 1 at the beginning, and so this is just equal to the covariance. I'm going to then substitute in what we said y was equal to here. So when we substitute that in, we have this. And then covariance is, I think it's biadditive. I think there's a term for it, but it's not, sorry? Bilinear, that's what it is. Which means that we can distribute this covariance through all the sums. And so, so there's going to be a bunch of covariances between xj and xk. And those covariances can be separated into the covariance due to linkage and covariance due to population structure. There's also going to be a term for the covariance between xj and this g. And then we've assumed that x is independent of the epsilon. And so this is what we have. This is what we have left. Now, in general, when we talk about the bias due to stratification, we're just referring to this part here. Why? But this is technically biased too. If we want to know what's the actual effect of SNP-J, then there's a lot of stuff in here that isn't the effect of SNP-J. Why might we care less about that? Thinking about like if the goal of GWAS is to identify regions that are associated with our phenotype, why is this bias not as concerning?
Because that can still be causal, just at a kind of less fine-grained way.
Yeah, what do you mean by less fine-grained?
I guess if you
Yeah, that's exactly right.
Looking up qualitatively, it's quite different because it's still causal.
Yeah, that's exactly right. So this term here, like because the correlation due to linkage tends to be quite local. It decays based on distance and is never across chromosomes. If we see SNP-J is associated, and we don't, we think that there's no bias due to stratification, we know that it's either SNP-J or a SNP nearby, and that's often enough to at least start thinking about, okay, what are the implicated genes, things like that. Whereas, population structure can cause correlation throughout the whole genome. And so if it's associated because of this term here, in fact, these ones here also are causal, right? It's tagging causal effects, but it's a causal effect any place in the genome. And so that's not very helpful. And then this represents just confounding that isn't due to any sort of causal effect. So that's not helpful at all. And so this term here we usually think of as the bias, and this is the actual kind of GWAS effect, unconfounded, due to population structure, even though it is, again, it represents the effect of a lot of SNPs other than specifically J. 1142. Okay, great. Oh, I move my hands a lot, and I'm worried I keep bumping the thing. Great. Okay, so next we're going to talk about, so I told you this is potentially a big problem. There's like maybe a lot of bias in our estimates due to stratification. And so how do we know if there is bias? Like how do we measure how much bias may be left? We do our GWAS, and we think, oh, there might be bias there. How can we tell if there is? And so there's kind of two main approaches, I think, to identify whether or not there is bias. I should maybe add a third one based on, well, I'll talk to you about the third one. Okay, so the first one is you can use within family data. So imagine that we have a sample of a bunch of siblings, and we randomly assign one to be sibling 1 and sibling 2, and we take the difference in the phenotypes of the siblings and the difference in the genotypes of the siblings. And so we're going to take our model that we had here before, and we're just going to take SIB differences, right? And so I take the differences, the y's and the x's and the epsilons. I don't have a delta g here. Why don't I have a delta g? Yeah, like I said that the g has to do with ancestry, and in principle siblings are going to have the same ancestry, at least the relevant ancestry part that we think is causing the differences in phenotypes between the groups. And so that term goes away, and so that's nice. And so we could then use this and do a little bit of math. And it turns out that once you do that, the within family beta that you'd get by running this regression, regressing the differences in phenotypes on the difference in genotypes, is just this term here. So it actually turns out to also cancel out all of these population difference effects if you go through like 12 pages of algebra, or maybe 6. And so that's nice. So now we have an estimator that has no bias from stratification. So why don't we just always do SIB difference models? There's just not enough data, right? Like, yeah, so family samples are just currently not big enough to do that. They're getting bigger and bigger. And so, we may be able to do like enormous, hundreds of thousands SIB paired GWASs like this within the next several years. But we currently can't really do it to get precise estimates for gene discovery. And so then what can we do with the SIB-based estimates? Well, we can take both of the sets of estimates. We can take our sibling estimates and our GWAS estimates, so the SIB estimates that are unbiased due to stratification, and the GWAS estimates which may be, and we can do some tests. So like one test we might do is a sign test. So if our GWAS results are 100% driven by stratification, and so there's no actual genetic signal at all, well, what that means is that beta within family is equal to 0. So what I'm under the assumption that all of the beta j's are equal to 0, then the within family betas should be equal to 0. And so the beta hats, the estimates from a within family analysis, is just going to be normally distributed about 0, right? Whereas the GWAS results are going to be normally distributed about just wherever the bias happens to take them. So that'll have random sign, just random based on the bias, where the bias happened to go. But it's going to be independent. It's going to have an independent sign of the within family betas, which are the sign is only dependent on like the sample that we happen to draw. And so we could look at how frequently the signs match in our within family estimates and our GWAS estimates. And so if it's true that there's actually no genetic effect, then they should only match 50% of the time, because it would just be by chance that they would match. On the other hand, if there's a true genetic effect, then it means that there's this common term. This term exists in both of them, and so it'll cause the sign concordance to increase. And so we have a pretty clean test here that will say, okay, is it true that all of our signal is due to population stratification? So we can do that. It's maybe a bit unsatisfying though, right? Because let's say that I find out, it's not all due to population stratification. But how much of it is due to population stratification? It could be that 1% is true signal and the rest is stratification, and that would be good to know. That would pass the sign test, but wouldn't necessarily mean that our results are useful. And so, 46. What time? I'm supposed to go till 12:30. Oh, we're good. Okay. So something that we could also do is a regression test. So let's say that we took our within family betas and we regressed them onto our GWAS betas. Okay, so remembering it's a simple regression. So it's the covariance of the two betas divided by the variance of, in this case, beta hat GWAS. Well, so the covariance between the within family betas and the GWAS betas, the only common term they have is like the true beta, right? So the covariance of those two things is going to be actually equal to the variance of the truth, assuming that they were estimated in independent samples. But down here, the variance of the GWAS has three parts. It's the variance of the true beta, there's the variance of the bias, and there's the variance due to estimation error. There could be some covariance terms in here. I'm going to assume them away. It's probably pretty reasonable to assume that there's not a covariance between the error and the bias and the betas. It's a stronger assumption to assume that there's no that the correlation between the bias and the true effect is zero. But we're going to do it anyways. So we have this function from the regression, but there's this term which is not very interesting. It's just a function of how big our sample size was, so we'd like to get rid of it. Fortunately, we know how big this is, right? So what's the variance of the estimation error? Well, that's what a standard error is supposed to give us, right? And so if we just use our standard errors and square them, that's the sampling variance, and we can just subtract that from the denominator, and we'd end up with an expression that looks like this. Okay, so this is maybe interesting, because imagine that there was no bias. There was no bias due to stratification, and this term is equal to 0. Then this thing should be equal to 1. So it means 100% of the signal is due to true signal. On the other hand, imagine this thing was enormous. So most of the variance was due to so this is really big. This thing would go down to 0, which means that approximately 0% of the variance that we see in our estimates is due to true signal. It's all being driven by the stratification. And so this estimator, you can think of it as the fraction of the signal that we're observing that's due to true genetic stuff versus stratification bias.
How do you calculate the bias due to stratification?
How do we do what, sorry?
Calculate bias due to...
Yeah, so we don't do it directly.
Oh, okay.
What we do is we just note that the variance of the GWAS betas is just going to be the sum of the variance of each of the pieces of the GWAS beta. So we say that the GWAS beta has true effect, bias, error, and we subtract out the variance due to the error. And so all that's left is going to be these two things. And so yes, but good question. Yeah, we don't directly observe it. We just know that it's there. Okay. Okay, so this is a way we can do these things. We can learn oh yeah, there is some bias in my estimates because of stratification. But I think I guess it's nice to know it's there, but it's better if we can fix it, right? And so let's talk about ways to address stratification that may exist in your sample. And so I like to think of it as kind of two classes of approaches that address stratification. So there are some that just adjust the standard error but don't do anything to your beta estimates. And there are some that actually will be procedures that cause your betas to be different. for things that adjust the standard error, let's talk about like what a standard error is supposed to represent, right? So a standard error, like in theory, kind of a high level, what we want it to be, is supposed to represent how much variation there is in our estimate that's due to stuff that we just aren't interested in, you know? And in most cases, the stuff that we're not interested in is just the sampling variance. So like we don't want, we want to know how much is our beta moving around as a result of the sample that we just happen to draw. And so, you know, for the most part, that's generally how we think of standard errors. But we could think, well, like maybe we'd also want the standard error to take into account the variation in our estimates that's due to biases like stratification. And so the idea of these approaches is that let's inflate the standard errors of our estimates such that for SNPs that don't have a true genetic effect, but only vary due to sampling variance and also maybe due to stratification, that, you know, we control the type 1 error for SNPs like that. So we only want to be able to, we'd only call a SNP true 5% of the time if the only thing that's causing it to, if the only thing that's making it like deviate from 0 is due to sampling variance and bias. So that's kind of the intuitive rationale of why you'd want to do this. The original way that this was done was something called genomic control. You don't see this done very frequently, and I'm only going to talk about it because you're going to see papers that do this. Like, you know, within papers that are even 5 years old, I think it was pretty common to see them doing genomic control, although I think that more recent, I don't think I've seen any paper that does GC anymore. But it's just good to know where it comes from. So the idea is, let's assume that, you know, we can take our SNPs and we can write them in order by p-value, just rank them all. And we can assume that the median SNP in our big list is null, that it has no real genetic effect. So when would it be reasonable to make an assumption like that? If what's normally distributed? If the effect of the SNPs are normally distributed, then the median effect, like every SNP is actually causal, right? And so we're saying that there are some SNPs that are null, and in fact the median SNP is null. Yeah?
If you have kind of incomplete polygenicity?
Yeah. So if you have a really sparse architecture. So if there's only like 10 causal SNPs in the whole genome, then the median SNP's probably also null. In fact, even if there's like 10% causal SNPs, the median SNP will also likely be null. And so this was the world that you know, when they thought of doing this method, that's kind of what people believed to be the case.
So just to clarify on the question, you have to think about taking the absolute value of the effect sizes?
That's right.
So a normal distribution.
Sorry. Yeah. Because the p-value has to do with the magnitude of the effect size and not the actual. Yeah. Thanks, Dan. And so if there's only a handful of SNPs that are causal, the median SNP will be null. And it used to be believed that that was kind of the case, that there were just a few SNPs with big effects affecting the traits, but and then everything else has no effect at all. And I think now we sort of understand a bit better that, you know, actually a lot of SNPs contribute to phenotypes with a very small effect. And so this assumption makes less sense now than it did before. But let's talk about what they do exactly. So what they do is they take the median SNP and they look at the chi-squared statistic of the median SNP. Now, if that SNP is actually null, then the chi-squared statistic of that SNP should correspond to the median of a chi-squared distribution. And if it's bigger than the median of a chi-squared distribution, they say, oh, well, that means that the SNP is more associated than I think it should be. And because I'm assuming it's null, it must be associated purely because of stratification. And so I'm going to inflate my standard errors until, so when you inflate the standard errors, you're decreasing the chi-squared statistic. And so you inflate the standard errors until the median chi-squared statistic, or the chi-squared statistic of the median SNP, is exactly equal to the median of a chi-squared distribution. And so you're just kind of forcing, you're forcing the distribution to be, to correspond to the assumption that you made. Yeah.
This is kind of a side note, but maybe it'd be helpful to explain why you're talking about chi-squared statistics rather than t-statistics, since people would think of that as the normal thing in a regression.
Yeah. But it was just easier. I mean, like it's. I mean, I think it has to do because there's a one-to-one relationship between, like, it's a monotonic relationship between chi-squareds and p-values, right? But for the same reason of like the t statistic, right? If you have a big t statistic, like big meaning big and positive, or big and negative, you know, they may have the same p-value, and you want them to be ranked approximately by their p-value. And so by squaring and make it into a chi-squared statistic, I guess it's f, but whatever. If you square it, then all of a sudden there's a one-to-one relationship between that statistic and the p-value. Great. So that's genomic control. LD score regression does a similar thing, although it relaxes the assumption of the median SNP having to be null. It allows for polygenicity. Raymond is going to talk more about that in his lecture, so I'm not going to talk about it now. But the principle behind inflating inflating the standard errors from the results of an LD score regression is based on the same principle. We want to inflate the standard errors so that they also account for the variation that may exist due to bias. I'm not going to talk about this. I'm just going to keep going. We can talk about QQ plots if you want later. Okay, so then let's talk about then ways that actually will change our beta estimates. Well, one way that we could make our beta estimates more robust and have less bias due to stratification is just by restricting our sample. So if we're doing our chopstick example, we would maybe just look at Europeans and try not to include people of Asian ancestry in our regression. And so that would, hopefully make it so that the G in the model that we had is the same for everyone in the sample, and therefore it will just get absorbed into the intercept, and so it won't be a source of bias anymore. And so this is why we say let's just reduce to just people of European ancestry, or more and more you'll have people let's just do people of Asian ancestry, or whatever you want to focus on. Great. In general, so the bulk of the data that has been collected and that people are analyzing today are of European ancestry, and in general stratification within European ancestors is small. It's not zero. In fact, there is some evidence of there's pretty strong evidence of north-south stratification in European samples. And so, the allele frequencies of people in Scandinavia are different than the allele frequencies of people in Italy, for example. And so even if you're using European samples, you need to be, you'll need to do other things to correct for stratification as well. I have a little point here that Dan kind of touched on as well. So stratification bias, when it's due to, under the assumptions that it's just kind of due to random drift processes. So like, okay, let me. There's different reasons that populations can be stratified, and kind of the two classes are, it may be that in one population a certain attribute is better than in the other, and so this is selection. And so if in some area it's better to be tall than in another area, then you might think that SNPs that make you tall become more common in that population. And so that's selection. That would be stratification due to selection. But I think most of the time we just think of it as stratification due to drift. Like if we had a big population and we built a big wall between them, then over time, because the alleles that get passed on is just random, you might have in one population the allele frequencies changing just a little bit by chance. But if we're separated by a big wall, then it's also going to drift over here, but it's going to drift independent of each other. And so they may end up in different places by chance. And so if that's the type of selection that's happening, this drift stuff, then the amount of correlation that exists between SNPs is evenly spread throughout the whole genome. And so the kind of bias that you would see is roughly constant throughout the whole genome. The expected bias at a particular SNP is the same no matter where you are. And so if we do our first GWAS and it has a really big effect size, we may be less concerned for our really big effect size SNPs because there might be some stratification bias, but maybe it's just small. But now we're getting million-person GWASs, and we're saying, okay, the betas for these million-person GWASs are much smaller than the original ones that we saw, but the biases are staying the same. are the same magnitude as we increase sample sizes. So what does that mean in terms of like how much of the signal is due to bias versus stratification when you have large samples? Yeah, the proportion is going to be much closer. And so even though maybe before, like as we get more data, we need to take the stratification more serious, because even subtle patterns of stratification can play a bigger role in our overall set of results that we're using. Mm-hmm.
Or for behavioral phenotypes that have smaller effect sizes relative to medical phenotypes.
Yeah, and so that's also true. So for medical phenotypes, where they just have much bigger effect sizes in general, you might be less worried about stratification, but we need to be extra careful. Even for our biggest effect sizes, they're more at risk of bias due to stratification. In terms of the proportion, if the effect is this big and the bias is just like that, maybe you can put up with that amount of bias. That's fine. Whereas if the effect is this big and the bias is that same size, then that's maybe more concerning. And so for our behavioral phenotypes, and then especially behavioral phenotypes where we're getting these enormous sample sizes, this is becoming a bigger and bigger problem that we need to take more and more seriously.
I think I might not understand part of the argument. You said that there is some covariance structure that you would expect if things are due to stratification, that is due to
drift.
Mm-hmm. As opposed to, the causal stuff. Let's put aside the selections.
Yeah, good idea.
That wall metaphor that you pointed out seems like inconsistent with the other metaphor that we usually carry out of like tree branching, and you could have different covariances associated with different times in which the population split from other ones and so forth. So I just didn't understand what the argument is, and is it at all sensitive to this different?
Alicia is here. She's going to make fun of my. It's like we have like this tree, right? And there's like branches that, like, is this the kind of thing you're talking about, right? Yeah. And so I think that even though sometimes these branches correspond to like physical geographic separations, right? So it could be that like these people leave Africa, and so now they're separated. There's not a wall, but that these people are just physically far away.
No. So what is the covariance structure that you were expecting if things are due to drift in different populations?
Yeah, so there's not something you necessarily expect, but you expect there to be differences. So the allele frequency over time, like even if you were, in a single homogeneous population where there's like random mating and everything is perfect, the allele frequency is going to roughly follow like a random walk. And so you have a bunch of allele frequencies all following a random walk, but once they split, they're following a different random walk. And so that may mean that there's mean differences between these two groups, because how the longer they've been separated, the bigger differences you may expect there to be. Mm-hmm. Does that answer your question at all?
I think I got it. I thought there was a more statistical, concrete structure that you expect due to drift.
I don't think that there's a structure where you like expect it necessarily to go up or go down. I think the statistics is that you expect you expect the differences to be larger on average when they've been separated for a longer amount of time. I think that's the, Alicia can correct me. Is that good? Or James? James has.
Well, I think what might answer the question is that there's genetic drift occurring at every single SNP in the human genome, and so the SNP on chromosome 1, allele A, drifts to high frequency in the population. And then also there's a SNP on chromosome 22 where allele A, let's call it, also drifts to high frequency in one population. Then there is, in the population is considered as an aggregate. There is a long-range LD, you might call it, between these two SNPs, because anyone who has A at the first SNP will also tend to have A at the chromosome 22 SNP.
Yeah, so that's how the correlations arise, and it's just due to chance if the frequencies happen to drift in the same direction in that population relative to the other one.
So in this population, one coin came up heads, and then by chance, another coin also came up heads. And then when you consider everyone jointly, the presence of heads at one will tend to predict the presence of heads at the other. But this is just due to, the stochastic fluctuations at these two sites.
Okay. Great. So yeah, so any other questions about that? Okay, cool. But yeah, the point I was just trying to make here is that we do need to be, and I'll probably make this point again later, so, you know, I guess you can ignore me now if you want. But we need to be increasingly aware of the risk of stratification. You know, just because we think that there's no stratification now or stratification isn't biasing the results that we're producing today, that doesn't mean that they won't be a problem 5 years from now for the methods that we're using that are using the new SNPs that we've discovered in the meantime. It's also true for methods, you know, so some methods just use the genome-wide significant hits, and some methods use SNPs throughout the whole genome. And so the ones that use the whole genome will also likely to be a bit more problematic for the same reason, because, you know, those SNPs will contribute small, like very little in terms of signal, and relatively a lot in terms of bias. Okay. So the primary way, and we've talked about this already a little bit, the primary way that we correct for stratification beyond just restricting our samples to those who have a common ancestry or have at least close ancestry is by using principal components. And so we're going to go back. This is the model that we have in our head when we're thinking about stratification. Now, if we observed G, like we knew the ancestry of every individual, we could just include it as a covariate. Or so these are the Z's that Dan talked about. If we include the Z's as covariates, that would totally explain all of the variation in G. And so there would be no bias induced by stratification then. So that would be the best thing that we could do if we knew everyone's precise ancestry. But we don't know that. And so, well, what can we do? Well, if you just think overall, when you look at a person's genome, like what's the primary thing that a person's genome represents? And I would say that thing is a person's ancestry. Like that's the easiest thing to predict if I see a person's whole genome. And if you think intuitively about what principal components is trying to capture, that's exactly also, you know, it's trying to capture what's the main source of variation that's determining, you know, what you see in all of your data. And so kind of thinking about those two concepts together, I think intuitively connects why PCs may be useful. If we take all of our genetic data and we take the first handful of principal components, those principal components will tend to be highly correlated with a person's ancestry. There is like a more careful mathematical rationale based on kind of simplifications of population genetic models and some random matrix theory. And so you know, we could be more rigorous about this if we wanted to. But I think that intuition is probably enough, and also just noting that this tends to work very well in practice. You know, including PCs tends to explain enough of the variation in G that it removes a lot of the bias. Yes?
Do you choose the number of components solely on the eigenvalue, or is there also some kind of biological?
I'm going to talk about that a little bit in a couple slides, if that's okay. But that's a great question. This is a great picture. We're going to show it to you again. But you can see that, you know, Europeans, I told you that it's a homogeneous sample, but nevertheless, you know, the first and second PCs are like approximately latitude and longitude. That's amazing. So we can just admire this a little longer if you want. Like, you know, here's Spain. I don't know. Okay, great. So we're going to actually get to your point right now. Well, it's going to be part of the main point. The main point here that I want to talk about is controlling for race versus controlling for PCs. So here's some data from the health and retirement study, and we picked some SNP in the health and retirement study, and we just did a GWAS, you know, we just regressed the phenotype. Is this education? We regressed education on that particular SNP with just age and sex controls. That's what's on the left here. All right? And so you can see there's a huge significant effect if you just control for age and sex. But then, like, you remember, oh, Patrick told me that there's stratification. I'm going to control for race because that's going to solve all of my problems. And you do that, and you do see a substantial decrease in the magnitude of this effect after controlling for race. And so you're like, oh, we're great. You know, I controlled for race, and now there's no more stratification. But you're wrong. What we've done in the rest of these points is we add PCs one at a time and we watch the evolution of these estimates as you add more and more PCs. So when you add the second PC, it decays more. By the time we add the third PC, it's no longer significant.
So that's kind of a scree plot for PCA?
A
Does
s-
it look different?
oh.
Maybe that's psychological.
It might be. I don't know what you said.
No, it's not. A scree plot would be plotting for each principal component the eigenvalue.
Right?
Oh,
That's
yeah.
not what-
This is kind of like that, but then with different parameters. Because you're looking at the drop.
No, because this is graphing the coefficient on the SNP in a regression with educational attainment on the SNP.
Okay. Yeah.
There's probably some link in between the eigenvalue and how much it moves, but I.
No, because the eigenvalue is just about the amount of variance in the
Oh,
genomics explaining, and this is
that's
about
right.
covariance.
The covariance between.
I think like how you interpret the drop in the sense of that you're looking for that drop, like you do in a script would be the same. But not the
Yeah, but kind of.
Yeah. So yeah, so you can see by the time we get to the third, it's no longer significant, and by the time we get to about 8, this seems pretty stable. It's like not really moving, right? And so this is you might interpret that to say, oh well, so maybe you need 8 principal components to get rid of the stratification. Another thing you could do is like, let's say you know the controlling for rate. Oh, do you have a question?
Yeah, should be this equivalent to doing like recursively within subpopulations that you are identifying? Because here you that's just adding, you take your pool of observations and then you say, okay, third, fourth, and fifth principal components are not moving this.
Mm-hmm.
But if you do the principal component analysis within subpopulation, you should be capturing more.
Yeah, within each of the groups.
Yeah.
Yes. I mean, so this is kind of what I'm going to show here, although I'm not going to do it within each of them. I think there's some additional assumptions that you're getting if you like actually separate it out, like you're assuming something about the interactions between the coefficients, but it's kind of the same sort of thing.
Isn't it assumed that their phenotypical kind of race or self-reported race perfectly matches ancestry kind of in a way if you stratify these?
Yeah, but you could take all of the people who are self-reported white and do a PCA among those people, and then you could take everyone who's self-reported Asian and do PCA just within them, and so you could do something like that.
I'm saying like if you do this first analysis, you will differentiate like some European population, but then if you do within France the PCA, you will find like regional differences.
Yeah, and there are people who are proposing doing things like that. So they'll do PCA, they'll cluster their PCA plot to try to find like groups, and then they'll do PCA within the clusters. So like, yeah, you could do that. I mean, the point of all of those things, again, is we want to get something that is highly correlated with this G.
Yeah, well, I'm just thinking like this G that is relevant will depend on what is your Y. So if you
Yeah, no, that's definitely true.
So if you are doing some behavioral trait that is highly variant, for other correlates with a group, and that group is like more fine-grained what you need, then just using the first 2 principal components in the overall population could be not as informative.
Yeah.
As just doing
Yeah.
recursively.
Yeah, I agree. And there are people who do things like that.
But eventually, so if you do principal components in whites, for example, and if you, after the first couple of components will identify north, south, and those big groups, and then when you go further, it's going to be sort of equivalent to doing it within subpopulations. So you're controlling for, when you include extra principal components, you're...
I mean, it's not exactly the same, but it is similar-ish.
Yeah.
I mean, you could come up with like a population genetic model where it does make sense to do the splitting and the PCAs with it. Like you'd get more information sooner that way. I'm going to show why it doesn't quite even work out then in just a couple slides. But yeah, sure, you could do things like people do things like that. The point of all of those things is you want something that will explain more and more of the variation in G. That's at least the rationale. Uh-huh.
Well, I mean, if you go back to this pretty image, I mean, I agree, it's pretty. There are like colors and whatnot.
Should we look at it again? Where is it?
Don't you think this is another version of winner's curse? And what I mean by that is, I mean, this is what made it to the Science or Nature paper, and it made it there because the separation is so quote-unquote neat. There could be hundreds of plots where this separation was
Doesn't work as well. Sure, sure. But what you could do is you could do, PCs, just so maybe this plot just happened to be pretty, just happens, and it wouldn't turn out that way if we did it again. But what generally is done is we'll include PCs and we'll have our results, and then we do follow-up analyses to measure how much stratification is left in our results using some of these things that I showed you before. And so in practice, at least so far, for most of the analyses we do, it looks like including PCs in this way is good enough for what we've been doing.
And just another related concern, because there's a lot of limitations associated with PCA, given it's a linear method. If you think about the history of migration, human history in general, don't you think any linear method in the world would be, too simple to capture all those patterns that neatly? It's too good to be true in a sense. That's my concern, because we have similar concerns in cancer genomics. Cancer is insanely complex, and sometimes you would see plots with two PCs every now and then explaining everything. And you just wonder, it's too good to be true. What do you think about that?
Yeah, no, that's a great point. So this is why we try to stick to homogeneous. So if we didn't have a homogeneous sample to begin with, so people that we think are European ancestry, and then we tried to account for all of the stratification with just PCs there, I think we would be unsuccessful for all of these complicated reasons that you're talking about. But once we restrict to a sample that we think is pretty homogeneous, so the UK Biobank, just all people who live in the UK, and then we do PCs on top of that, it appears to be pretty good.
All right, if you don't mind, I have a third concern. Because for countries, especially like the UK or the USA and whatnot, they're far from being homogeneous. Even on top of that, how people identify their ancestry, there's no single definition of ancestry.
Yeah. So this is not self-reported ancestry. We determine ancestry based on using PCs. No, but you look at the pictures, right? If you were to do a PCA plot, you normally see there's usually a big cluster here, and there's a smear here, and I think, and then what we do is we just take the people who are in a very small region of the PC plot.
But Patrick, if I send a paper to Nature Genetics with super noisy clusters, will they publish it? Probably not.
But why do you think they would be so different? The PC plots always look alike. There might be some noise, but they don't look randomly.
The published ones, that's my point.
No, if you do it yourself, try to do it yourself.
I probably will, but the whole point is we always see what comes out in a publication, and it might be the case in certain scenarios, but there's this publication bar that things should.
So Alicia is probably the expert in the room on these kinds of. So Alicia, how consistent do you think these PC plots are across data sets?
So it kind of depends on the composition of the sample. And looking at something like Opera that picked the plot from John McNevray, those individuals were selected for having 4 grandparents born in the same country. So those individuals are going to look more like they fit into clean, consistent little bins on a PC plot than something that you get in the US. In the US, you'll have lines of ancestry, say.
So a Klein is like the schmear, where it's
More of a smear, exactly. So they're not going to use clean, discrete bins, but that's fine. We don't have clean, discrete bins of ancestry, and that's what we're trying to correct in our. That's the signal we're trying to correct in our data. Is this continuum of structure. I don't necessarily know what to add to that.
Yeah, so I'm just, I think that the big point that I'd like to make is sure, I take that ancestry is complicated and not linear and is messy, which is why I say that even when we're doing all these things, we need to be very conscious about how much residual stratification may exist in our data, and we should test for it if we can. In practice, it seems to be working. But in your application, you should check as well. Just because it's worked in the data sets that I've worked in, that doesn't mean it'll necessarily work in your data set. And so check. Do the things you can to correct for it, and then do these tests to make sure that it also has worked.
I guess one other thing to add to that is that when you have heterogeneous population and you have a massive proportion of your data coming from one ancestry group, your PC space is going to be warped towards that ancestry group. So it's going to distinguish more of the variation within the bulk of your data and less of it
Yeah, I'm going to actually talk about that on the next slide. This is just illustrating that if we restricted just to whites, so we told you that you shouldn't just control for race. So we say, okay, well, let's just restrict to whites, and you see the same sort of thing. So even when you just have self-reported whites, there's a lot of bias that exists that can, eventually seems to be controlled as you add more and more PCs. Yeah.
Is the horizontal line, just to clarify again, is the horizontal line like a legitimate zero regression, or is that zero difference?
So the dotted line is 0.
Okay.
Yeah, the horizontal line just means that we've connected the dots, and so it just it's just to show a little bit that.
The dotted zero line is a legitimate zero regression coefficient as opposed to zero difference from the within.
Oh yes, that's what it is. Mm-hmm. Yeah. Okay, so let's talk about there's a big GWAS of height, Wood et al. 2014. And that was kind of one of the biggest GWASs when it came out. It was a big deal. They found lots of hits, and it's been used for lots of follow-up research. There were 2 papers though that came out this year. Actually, they were in the same journal, and they came to the same conclusion that the results from the Wood et al. height GWAS seemed to have lots of residual uncontrolled population stratification. And so this made a big splash, and people are thinking, well, why? They tried to do all of the state-of-the-art things when they did their analysis. So why is there so much stratification left in their data? Well, there's a number of reasons. So one of the first ones is this was a big endeavor. There were nearly 80 studies contributing to this meta-analysis, and I think as a result, QC is annoying and time-consuming and very hard, and we love ISU so much. And so it's possible that there were errors that just crept in because the analyst made small mistakes that weren't caught by people down the line. Not even necessarily because of neglect, right? Like when you have a lot of the QC metrics that we have are statistical tests, and when you have a lot of studies and some of these studies are very small, these QC tests are underpowered, and so there may be a problem, but we just don't have power to detect the problem. And so, so this is maybe one reason. There was bias that existed in each of these small studies that just remained when they meta-analyzed them together, and they couldn't catch it in the first round. There's maybe another issue. The primary way that they controlled for population stratification in these studies, they did PCs, but they also did a lot of genomic control. Genomic control doesn't affect the betas. It just inflates the standard errors, right? And so if the bias was systematic for the things that people wanted to test in follow-up, you may think that each of your betas are, the standard errors for each of your betas may be all right, but the bias will compound when you take like an aggregate test for selection or some of the things that people were trying to do with this data. But the third point I think is actually, I think is the primary reason that there were problems. And the issue is that PCs have to be estimated, right? We estimate it based on the data. And we could think that if we kept drawing from the population, there's like a true PC that may exist that we would get if we drew a sample of infinite size from that population. But what we're getting is a noisy, an estimate of that PC. And PCs have a few funny attributes. So here's actually an unpublished paper by some of my colleagues at Broad. And so what they have plotted here is on the x-axis is the sample size that you're using to calculate the PCs. And on the y-axis is the correlation between the estimated PC that you get in your data and the true PC that you would get if you had infinite data. And so, you can see for PC1, sorry,
What is true PC? There's true ancestry, right? But there's no true principal.
Well, there kind of is, right? So let's say that we kept drawing from the sample, like over and over and over again to an infinite size. The PCs would eventually converge to a particular value, right? So because PCs are just weighted sums of genotypes, like you can characterize them that way, and so those weights would converge to a specific thing, which means that you could calculate a person's like actual PC, that like an unnoisy PC equal to the thing, because it's that true thing that in theory should be correlated with ancestry.
Yeah, but I just like theoretically, it's hard to understand what a true PC.
Yeah, and it's a bit weird, but what I mean by that Yeah, I mean, yeah, what I mean by that is the PCs that you would arrive, if we drew an infinite sample, then over time, the PC for a specific individual would stabilize, and it just would no longer keep moving. And so when I say true PC, that's what I mean, is it's this value, and ultimately that's the value that we're interested. The reason that PCs control for ancestry is because this true thing that it converges to is the thing that we think will be correlated with a person's ancestry. But it is kind of a weird concept. You're right. Yes.
That's true. Is that not only true in the sense that in the PC you're treating all variation as maximally informative, right? You're explaining variances rather than covariances. So it's qualitatively different to have differences based on population stratification due measurement error, or some of the things you alluded to earlier. Like actually captured heterogeneity within the measurement itself rather than true variation in population.
I don't quite follow your question.
So is there any chance that part of the reason why your PCs converge to not adding much more is that by that stage you're just essentially explaining measurement error?
Yeah, no, and that's the point that I'm going to make here. That's exactly right. So for the first PC, this is capturing where the maximum variation exists, and so even in small sample sizes, we get a pretty good estimate of the first PC in a small data set. But the second PC, even with 2,000 individuals, the correlation of PC2 with like the true PC2 is 30%. And the third PC, you can see it's pretty close, like even in the larger sample it's small, and down here it's close to 0. And an interesting point that they point out in this paper that I think is fascinating is that these aren't approximately 0, these are exactly 0. So PCs have a phase transition that until you have a large enough sample size, the correlation of your observed PC and the true PC is exactly 0. It adds exactly no information. And the reason that this is important is because you'll often, just one second, James. You'll often see people say like, oh, I want to really make sure that I'm controlling for all of the stratification. I'm going to include 100 PCs, right? And like that sounds nice, and you think like, yeah, the 100th one maybe doesn't contribute much. But like, it contributes a little bit, and no, it doesn't. It contributes exactly 0. It's 100% noise. Go ahead.
Well, how can we be sure that it's just the PCs have switched order? That their eigenvalues are slightly fluctuating as the sample size changes, and so PC2 becomes PC4 in the next?
That's a great question. And I don't know how they address it in this paper.
Okay.
I think they're going to have a manuscript that they'll post online very soon, and it would be very interesting. Yeah.
So practically speaking, what you would do, if whenever possible, if I understand correctly, if you're a small British cohort, say, then what you could do is to fix this problem is estimate the weights in UKB.
Yeah.
Some sample of UKB that you think is representative of your population, and then this would go away.
Yeah.
Principle.
And that would be, well, I mean, it wouldn't go away, but at least you could get probably a lot further and have PCs. Oh, I should.
Closer to the measure the true PC is much more accurate.
That's right.
And some cohorts do that, but it's certainly not the predominant procedure.
Mm-hmm. Yeah, I mean, it's something you may also, you may consider doing, if you have a small cohort, I mean, you may get better, there's a trade-off here. You might get better traction calculating your PC weights in a very large sample that isn't your sample, and then using those to your PCs. So on one hand, your weights may not exactly represent the population structure in your data, but on the other hand, you're going to have, there's a bias variance trade-off. You'll have population, the SNP weights will be a little bit biased because you're using the wrong populations, but you're going to have the much more precise estimates, and so maybe it'll be a little bit better. So this is like cutting edge, brand new, interesting stuff. I don't know that I have, there's not a status quo way to do this kind of stuff, but it is something that I think is interesting and new and important to keep in mind. Yes?
Just one question. What is the small sample size? Because
Well,
compared to GWAS, everything is small, in individual studies.
Yeah. I mean, so in this case, what sample did they use here? I don't know which sample they used, but in this case, 2,000 people. With 2,000 people, your PC2 is already not great, and your PC3 is really bad.
For mental health research, 2,000 people is a lot.
Exactly. So that's what I was saying. You may consider, if you're doing a small study like that, you may want to consider calculating the SNP weights for your PCs in a different sample drawn from the same population, and then using those.
I wonder if you should do both, though,
Oh.
because I mean, there's also going to be issues within your particular sample that you probably want to capture.
That's actually true, especially if it's like, yeah, that's true.
So.
Yeah, so there's biases that may just due to the fact that half your sample was genotyped at one time and the other half was due at another time, and those will also be captured by PCs, and so that's a good point. So you could have both, but the ones that you do in sample will unlikely capture more than just like super basic levels of ancestry. Mm-hmm.
It also doesn't hurt to control for 5 or 10 PCs, right? Because you just lose a degree of freedom for each one you add.
Yeah.
1,000 people, that's probably not a big deal.
It's not costly. It's not very costly to add them. It's not going to cause problems. But it's not, But adding 100 PCs isn't going to make your results any more credible than 15. Mm-hmm.
So after the fact, could you go back and say, like, imagine that you found certain highly relevant loci for your GWAS study, and then you just say you assume the fact that there are some members of the population who have sort of mixed ancestry potentially at that loci.
I might have run out of batteries. That's okay. We're out of time anyways.
So I was just wondering if, like, you could go back after the fact and actually look at your samples and then compare particularly like high GWAS samples and look at them and see if they're distributed across those clusters.
High GWAS? Is that what you mean?
I mean your highly significant loci.
Mm-hmm. Oh, and look at how like.
Frequencies differ between ancestries, I guess.
Yeah, the distribution across your PCs after the fact. It's just another way of making sure that you have a good distribution rather than focused clustering.
Yeah.
Checking whether they're ancestry informative SNPs.
Yeah. I mean, I guess so I mean, if you've included your PCs as controls already, though.
Well, the reason I asked that is the following. Like, I imagine that if you had, say, an individual who comes from mixed ancestry, so the PCs, what they're essentially doing is they're correlating across all of the genotypes. So if there's a section, say, a recombination section in their genome, something like from a small set of loci, that could be washed out by the overall correlation structure, right? Because it's a small region. So my point is that after the fact, if you go back and try and do an analysis on that, maybe you could pick up those fine-grained kinds of mixed ancestry portions after the fact.
Yeah. I mean, hopefully, so when you say mixed ancestry, again, we're restricting, so we're like northern, southern England?
Well, kind of. Well, in that if you label as one, but you tend to have some mixed ancestry from the other, just even if you do a binary 2-cluster kind of thing, right? North, south, whatever you want to talk. I'm just saying that the fact that you could have a little bit of mixing of that, that's all.
Yeah. I mean, it seems like that I mean, it seems like a reasonable thing. The only reason that I wonder if there's much information is the fact that you've controlled for it already. But sure, I mean, there might be some information there. Mm-hmm. I mean, we're already 5 minutes past the extended time. I was going to talk a little bit about mixed linear models, but we should maybe have lunch. I don't know. What do you think, Dan?
Maybe just say a minute or.
Okay, I'll say a minute. Okay, so something to think about. So when we control for PCs, imagine a procedure where what we do first is we do a model where we regress the phenotype just on the PCs, so no SNPs, just the PCs. And so then we create a predictor for our phenotype with the PCs, and then we residualize out the variation that's due to those PCs, and then we would just regress the residual onto the SNP. Now that's approximately equivalent to just having controlled for the PCs, right? And so we could think of controlling as sort of like predicting in these other groups. And so there's a class of methods in genetics called mixed linear models. Okay, well let me say this first. So why do an OLS predictor using the PCs? We could do something that's better at prediction, like Ridge or Lasso, and we could use all SNPs rather than just like a handful of PCs to create the predictor. All right, so that's something that we could do, and maybe that would be better, because this predictor would absorb the variation that's due to the bias and residualize it out, and so all that's left, hopefully, is like the true signal that exists that we're getting in our regression. And so very briefly, there's a class of methods called mixed linear models, and they do approximately that. When you read papers on mixed linear models, they don't actually say they're doing this, but they are. At least like Bolt LMM is one of the ones that's pretty popular, and that's what they're doing. They'll take all the SNPs on other chromosomes, like so you have some SNP you're interested in. You take all the SNPs on another chromosome, you predict using some sort of fancy Bayesian something or another, and you residualize it out and then regress on that. And so the idea is that all the other SNPs are a random component. So you could treat a prediction algorithm as you're treating all the betas as random. There's an interpretation there, and you're residualizing that out, and then the beta corresponding to SNP you're interested in is the fixed effect that you're interested in, which is why they call it a mixed model, because there are some components you're calling random and some components you're calling fixed. And so this seems to be pretty effective at controlling for population structure as well. And the connection to the PCs is exactly that. It's kind of equivalent to doing this prediction and residualizing it out. I think that's all I want to say about that. So concluding thoughts, stratification is important, and the importance of stratification changes over time as we get increasing sample sizes. So I don't think we're ever going to get to the point that we say we've solved it. It can show up even when you think your sample is racially homogeneous or even homogeneous by ancestry. There's lots of tools that you can use to measure and test for it. We talked about a bunch of them. And again, this point that I said again, as we get more and more precise and we're trying to identify smaller and smaller effects, stratification becomes a bigger and bigger problem. Great. Thank you. Thank you.